להפיל את זה כל פעם עליך, פשוט... Okay. So, last time we were in the middle of talking about G-protein coupled receptors, and the lesson before we talked about in general signaling molecule and cell surface receptors, and that G-proteins are one example of this of a type of signaling cascade or signaling molecules. And specifically, this is like a basic structure of a G-protein. You have the G-protein coupled receptor that associates upon activation to a G-protein. That the G-protein has three subunits. The alpha subunit, which is GTP-as, meaning that it hydrolyzes GTP, that gives the energy for the conformational change that it needs to do, and the beta-GABA subunits that normally dissociate from the G-alpha upon activation. We showed a very schematic view of the activation of the G-protein, the components, the receptor, the G-protein itself, and the effector protein that the G-protein activates. And we described the basic steps of activation when we said that, for example, one hormone can activate many G-proteins and thus activate many active effector proteins. So there is a level of amplification also in this level. And we didn't talk about the second type of G-proteins, but I just mentioned for you so you won't be left in the dark. So that there is also a second type of G-proteins, which are monomeric. All the G-proteins that we talked about and we're going to talk about are trimeric G-proteins, but there is also a subclass of monomeric G-proteins. And the difference is that they associate with the receptor in a non-direct way. So they have like this adapter proteins that they associate with with the receptors through. This is like the molecular representation of the switch compartment, or the GTP binding compartment of the alpha subunit of the G-protein. And there's like this confined or stressed conformation that the protein assumes upon association with the GTP, and this is what we call it. We call it switch, and this is the on state of the switch, and this is the off state where it's bound only to GDP and not GTP. So I also mentioned that there's a few types of different alpha subunits. The difference between them is that some of them can inhibit their effector protein, some of them can excite their effector protein, and so there's about 27 different types of G-alpha proteins, and this is when the book was written, so probably there are more by now. And you also have 5 G-beta and 13 G-gamma subunits. Each one of the different combinations of them gives a different activation of their target effector proteins. So we gave the example, the classic epinephrine example, which is part of the body response to stress, or the body response to fight or flight mode. And we said that 3 things happen that are mediated by G-proteins in the response to epinephrine. First, the catabolism of glucose and fatty acid that produces more ATP for the body to, or for the cells in the body to have more ready energy. This happens through beta-adrenergic receptors. Then an increase of heart rate contraction rate happens also through beta-adrenergic receptors, and cutting off circulation of like the stomach and kidney happens through alpha-adrenergic receptors, but all of these receptors are activated by epinephrine. So beta-adrenergic receptors, which is the example here, can activate an activator G-protein that will cause an activation of an NAC class and produce elevated amounts of cyclic AMP, which is the secondary messenger. And alpha-adrenergic receptors that are coupled with G-inhibitory or GI proteins can inhibit that NAC class and produce less cyclic AMP. So one type of receptor can inhibit the production of cyclic AMP, and the other one can activate the production of cyclic AMP. Obviously, sometimes these two reactions will compete within each other in a given cell, but but specifically, many times you will have a separation of these two. Like one of them will happen in one cell, and the other one will happen in a different cell, but still it's in response to the same. It can happen in response to the same hormone. So after we have the cyclic AMP, what it does, it activates protein kinase A through binding of the R subunit of the inactivated pKa, releasing the catalytic site, or releasing the activated C subunit of the pKa, which can then phosphorylate and activate more proteins in this pathway. So again, we said that there are four main levels of amplification that the G protein gives us. First of all, one epinephrine can cause, through the activation of multiple G proteins, can cause activation of multiple adenine cyclase effector proteins. These adenine cyclases can, each one of them can produce multiple cyclic AMP molecules. And here there's no level of activation because the cyclic AMP, two molecules of cyclic AMP are enough to activate one protein kinase A. But then protein kinase A can activate many enzymes, and the enzyme can produce many products. So there is a very high level of amplification from a single epinephrine molecule. But if we compare this response relative to what we learned about neurotransmission or ionotropic receptors, stuff like that, this is much slower. Okay, so and the normal termination of the signal, like I'm talking about normal circumstances, not like very high signal, but under normal circumstances, the affinity to the termination of this signaling cascade will happen either by the affinity of the receptor to the hormone will decrease over time because of the breaking down of the G protein, or an enzyme that will break down the cyclic AMP from its cyclical form to a linear form, and will terminate the cellular response. Now, this is under normal conditions, and under prolonged activation, for example, if you have let's say you're stressed for a very long time and you have a release of a lot of epinephrine all the time, then the body will start to desynthesize, or the receptors will start to desynthesize, because you don't want to be in like major stress all the time. And one of these examples is through an enzyme that's called beta-adrenergic receptor kinase, or BARK. And the BARK is activated by pKa, like we said before, that is activated by the hormone pathway itself. And what BARK does is that it can phosphorylate the adrenergic receptor in the cytosolic segment, or cytosolic portion of this protein. And this phosphorylation actually causes a reduced affinity of this receptor to the G protein itself. So this phosphorylation is like, it does two things. First, it like acts as you could say acts as a time constant keeper for this response, meaning that it takes about 10 minutes. Like this phosphate will stay on the protein for about 1 to 10 minutes. So if the response like decayed, this phosphate will eventually be removed, and then the receptor will return to its normal sensitivity rates. But if this prolonged activation continues, then you will get more phosphorylation, eventually leading to a binding of a molecule that we said it's called a binding of a protein that's called beta-arrestin. And this beta-arrestin will completely block the ability of this receptor to activate the G protein and completely desensitize this specific receptor, even in some cases leading to endocytosis of this entire complex with the receptor and removing it from the plasma membrane of the cell, so causing like a more long-term desensitization. Okay, and this is, I think, ano. Then we move to the next example, which is G protein coupled receptors that regulate ion channels. The most simple example is through like vagus nerve activation of muscarinic receptors through acetylcholine. So the muscarinic receptor of acetylcholine can activate once it's bound to acetylcholine, introduces a conformational change, binds G alpha, and in this case, the the subunit that actually does the work is the G beta gamma subunit that binds to a potassium channel and causes it to open. And once this potassium channel is opened, then the potassium leaks out, and the cell becomes more hyperpolarized. And if the muscle cells are more hyperpolarized, then it causes the heart level to reduce. Like the speed of the pumping of the heart is reduced due to this activation with acetylcholine. So, and the last example that we started showing you is that is the one of the visual system and the rhodopsin. And the rhodopsin is a type of G-protein coupled receptor, but in this case it's activated by light. Okay, and you know, one of the things that's nice about this course is although I know that a lot of the students learn most of the material when they study for the exam, because this is when you go over it for real. For me, it's a continuous learning process, and I learn all the time and get new insights, so it's very interesting for me, which is important. And one of the things that I realized when I prepared this slide is that actually I never thought about why rhodopsin cells have these multiple discs, because it looks a bit like a strange structure. And actually, when you think about it, this is one of the classical examples of where the cell or the biological system can take like a system that is already working somewhere else and saying like, okay, so we know that the G-protein receptors are dependent on a membrane to be activated or to function, because the entire thing relies on diffusion, on lateral diffusion of the elements along the plasma membrane. So it's like the biological system said in this case of the rod cells, say, okay, so we don't have membranes inside the cell, so let's create an organelle, a special type of organelle, which are these membrane discs. And once you have these organelles inside the disc, and you have multiple organelles or multiple discs inside each rod cell, then now you can use the G-protein coupled receptor system to detect light. Okay, so it's like there's probably, like, I don't know how it really happened, but it's one of the, I think, the nicest example of like a use of a known biological pathway or a biological pathway that works somewhere else and adapting it to a different function, which is something that we see in biology all the time. So in this case, there's no ligand, and we said that the ligand is actually, we can say that it's already bound to the rhodopsin itself, and the ligand is a retinal molecule. And this retinal molecule, when light is absorbed by this retinal molecule, or a photon is absorbed by this retinal molecule, it changes conformation from cis to trans. And this conformational change is like a ligand binding that we saw in previous example. It causes a secondary conformational change in the rhodopsin protein itself. And this conformational change is what allows the G protein to bind to this receptor and start the entire cascade. And I will not repeat the example that I gave you before, but this is an approximation of a crystal structure that we have of the rhodopsin together with the G proteins, or the G protein that binds to it. Again, you can see the G alpha and G beta gamma subunits. And the operational mechanism is that during dark, the rod cell's resting potential is minus 30 due to the fact that you have constantly open channels that allow entrance of sodium and calcium inside the cell. So the cell is like always depolarized constantly and releasing neurotransmitter constantly to the receiving cells. And this is because of cyclic GMP that is again under dark conditions. It has high cytosolic concentration, and this high cytosolic concentration allows it or causes that many of these channels that are dependent or gated by cyclic GMP will remain open. Okay, and this is what causes the cell to be depolarized all the time. So once light is absorbed by the opsin, then the retinal molecule inside undergoes this conformational change. The opsin itself undergoes a conformational change, and the entire cascade that we talked about before also happens here. The exchange of a GTP molecule instead of a GDP, and this alpha subunit now laterally diffuses along this disc membrane. This time, this is not the plasma membrane of the cell, causes activation of an enzyme that's called cyclic GMP phosphodiesterase. So what this enzyme does is that it breaks down cyclic GMP to its linear form, which is GMP, and GMP cannot open these channels. And because now we have low cytosolic concentration of cyclic GMP, then these channels close. The membrane potential returns to a hyperpolarizatory state, and it results in reduction of release of neurotransmitter. Okay? Yeah.
So the opsin is the receptor, right?
Yeah.
And it's permanently bound to the retinol, or do we consider the retinol part of the receptor system?
It's permanently bound. Yeah. They're covalently linked to one another.
They're two separate things.
I think it's not. I think that the retinol is something that is coming from metabolism. It's not part of the protein, like to begin with. When the protein is transcribed from, like when it's translated from mRNA, and when it's produced, it doesn't have a retinol originally. This is something that is added to it later. So I think that's why you separate between the two. Like the retinol is not encoded in the genome, for example. Like, but the binding happens later. Okay. So again, we said that this process seems like, when you think about vision, it seems very, very slow, right? Like for this entire cascade to happen, it takes about, this can take up to 100 or 50, actually 20, I think, yeah. It's up to 20 milliseconds it takes for this thing to happen, which is very slow in molecular terms. But what it does is that it gives us the ability to detect close to a single photon. Okay? When the system is fully sensitive, we have the ability to detect a single photon, because a single photon can elicit such a massive response in the cell that we can interpret it, and in biological terms, it probably is more beneficial to be able to detect something and then to be able to detect something very, very fast. So one of the things that's amazing about human vision, and this is something that is pretty unique to humans, is that we have a very, very large spectrum that we can detect light, or levels of sensitivity where we can function under like varying lighting conditions. So a human being can see pretty okay, not the best. Like one of the things that people say that is unique to human beings in general is that we're not the best in terms of sensory. Our sensory system, we're not the best at like specific functions, but we have a pretty okay sensory system for all the different types of functions. So you have, like, for example, animals that can see better than humans at night, but they don't have color vision. Okay, so it's like, so humans are like okay in all terms, but they're not the best in anything. But so this is one of the reasons probably that humans are successful. So if you're not the best in what you do, then maybe it's okay. You need to be versatile. So the rod cells specifically are activated, are what gives us a black and white vision or low light vision. So from here on, it's different types of cells that we're not going to talk about. They're cone cells, and they're responsible for full color vision. But in these lighting conditions, the rod cells are the ones that are active, and the rod cells themselves have a very high capacity to adapt the response to varying levels of ambient light. Okay, so I showed you before the example that it can detect a single photon, but what happens when you're like driving at night, when you don't have like full sun, but it's like somewhere in the middle? So the way that they adapt to varying levels of ambient light is either by receptor phosphorylation and cellular translocation of G-proteins and arrestins. So the receptor phosphorylation happens in a very, again, think about the fact that we're talking about visual system, and before we described an adrenergic response, but still you can see that the cellular components are very similar to what we saw before. So the opsin, or the receptor phosphorylation, happens with an enzyme that is very similar to the beta-adrenergic receptor kinase that we saw before, that can phosphorylate the opsin. And each opsin has 3 principal serine phosphorylation sites, and the more sites that are phosphorylated, the less able is the activated opsin to activate the G-alpha. Okay, and thus causes it to be less sensitive to light, in essence. So because the number of photons that you need to activate the same response that you had before, you need much more light to activate or to elicit the same cellular response that you had before. So again, after like in dark conditions or in very low light settings, all your opsin proteins or opsin receptors are unphosphorylated, meaning that every photon of light that will be recepted will elicit like a full-blown cellular response of the cyclic GMP and the G-alpha and all the things that we saw before. And and this is what will happen here. So you have a massive amount of activation from the activated opsin. Then, when lighting conditions become higher, the receptor starts being phosphorylated, and its ability to activate the G-alpha is diminished until in the last phase, if the entire receptor is phosphorylated, similar to what happens in the example that we saw of the epinephrine, an arrestin molecule can bind to this phosphorylation site and completely block the activations of G-alpha. So this normally happens under full light conditions, and actually your rod cells become completely irrelevant, and you entirely rely on color vision, for example. In this case, I don't think that there is the following endocytosis and the long-term effects don't happen here, because otherwise we would have very hard time to adapt to different lighting conditions, obviously. But still, when you think about this, you can understand what happens, for example, why it takes so long for you to adapt to dark condition. It takes like about 45 minutes for you to adapt to full darkness. And part of it is because that you need time for these phosphorylation sites to clear and to regain the sensitivity of the system to what it was before. So this is one type of mechanism that helps the system be sensitive to varying levels of light. And the second one, which is a more long-term, is the cellular translocation, or the cellular location of the proteins that are involved in this signal cascade. So if we look at this rod cell, it's divided into compartments, and only in the outer segment of this cell will you have these organelles or these discs that contain the opsin the opsin receptors. So this is an image like a side section of a mouse retina that was taking that. In this case, they stained, and this is like a very crash course in immunofluorescence. Is that if you want to see a specific type of protein, you can target an antibody. Normally, you target first a secondary antibody, a primary antibody that knows how to bind to that target protein that you want to see, and then you target a secondary fluorescent antibody that targets the primary antibody, and then you have a fluorescent view of the protein that you're interested in. So in this case, they put a fluorescent antibody against arrestin, and in this case, they put a fluorescent antibody against the G protein. So what you see here is the location of the G protein and the location of the arrestin along the cell under dark and light conditions. Okay, so the small spots that you see here are actually, because this is not a single cell, this is the entire retina. So in the entire retina, you have a very compact array, but a very organized array of of these rod cells, and but all their outer segments are located like in the same retinal layer. So one of the things that we can see that in dark conditions, there are almost no arrestin molecules in the relevant compartment of the cell, meaning that it will take that there will be no inactivation of the receptor even if there is phosphorylation. Okay, so it's kind of the way that the system like retains the highest sensitivity under dark conditions, and like you can see also the G proteins, which are important for the cascade, are highly abundant in the relevant portion of the cell in dark conditions. And in light condition, the arrestin completely reverses, like it migrates to the outer segment of the cell, and in this case, it will, because the high concentration of arrestin, any phosphorylated protein will be rapidly targeted by the arrestin and completely blocked. And the G protein, like, kind of diffuses along the entire compartment of the cell, and is relatively lower concentration in the outer segment. Okay? Questions? No. Okay. So, the last example that we're going to talk about the G proteins is the activation of gene transcription by G protein coupled receptors. So, one of the nice examples is that is one of the genes that people discovered, which is called TABI, and this time it's a mouse discovery. So it's funny that they gave it a nice name. I told you that Drosophila people normally give cool names, but this time I don't know. The mouse people probably studied Drosophilas before. Anyway, so this gene, or this protein, normally under normal condition is bound to the plasma membrane, to the inner leaflet of the plasma membrane, through this lipid anchor. And what happens through activation of of signals that are coming from the related, if you know about leptin or other molecules that are responsible for feeding behavior, activate the type of receptor that activate G proteins, that these G proteins will activate an enzyme that's called phospholipase C. The phospholipase C will cleave this anchor from the TABI protein, and the TABI protein can now diffuse inside the nucleus of the cell, bind to DNA, and cause transcription of genes that are related to feeding behaviors. And for example, mice that this TABI gene was knocked out develop massive obesity. I don't know if you've seen mice, but this is not how normal mice look like. And the second example, which is a very classic example that you already saw in the Candel paper, in which a G protein coupled receptor activates the DNA class, increases the level of cyclic AMP, the pKa then gets into the nucleus, phosphorylates CREB, cyclic AMP responsive element binding protein, and this phosphorylated CREB can now bind to Cre, and through mediation of mediatory proteins can now cause the transcription of certain genes that are responsible, for example, for plasticity, long-term plasticity like we saw, and other behaviors in the cell. But this is not restricted. For example, this response is not restricted to neurons. It can happen in many different types of cells and cause transcription of many different types of genes. Okay, so how do we measure, or how do we quantify the speed or the activity of G proteins? So first of all, we need to understand like very few, like a very basic concept, which is fluorescence. So fluorescence, before we understand what is FRET, which is the second method. So fluorescence is a quality of specific molecules that once they absorb, or this is in the atomic level, it's actually described. This actually describes two different energy states of an electron that can absorb a photon of light. When an electron absorbs a photon of light, it can switch from a low energy state to a high energy state. then radiate, and eventually when it drops down from the higher energy state to a lower energy state, it releases a photon, and normally this photon that will be released, will always have lower energy than the photon that was observed. So if a photon that moved the electron from this level of this energy level to the higher energy level of was, for example, a wavelength of 400 nanometers. So I don't know if you know, but shorter wavelengths have higher energy. Okay, for example, X-ray, which is very, very short wavelength, is a very high energy radiation relative to, let's say, infrared, which is a lower level of energy. So in this example, if we have we have a special protein which we call a cyan fluorescent protein. This protein, if we excite it or we shine a laser on this protein that is in wavelength of, let's say, 420 nanometer, what it will do, it will emit a photon, or it will emit fluorescent energy in the wavelength of about 500 nanometer or 488 nanometer. Okay, so how exactly can you use these systems in order to measure G proteins? This is done through a physical phenomena which is called Foster resonance energy transfer, or FRET, which happens when two fluorophores or two fluorescent molecules are in very, very close proximity to one another, meaning that less than 10 nanometers in proximity from one another. And when they're very close to one another, what can happen is that when one fluorophore will absorb energy, instead of emitting a photon in lower energy wavelengths, the energy will transfer from one fluorophore to another fluorophore, and you will get an emission in the emission wavelength, or the normal emission wavelength of the other protein. Okay, so again, in this example, we have two types of proteins. We have cyan fluorescent protein that when we will excite it with a laser with 420 nanometer, it will emit around 500. And we have another type of protein which is called yellow fluorescent protein, which is YFP, in which if we excite it with a, let's say, 500 nanometer laser, it will emit in, say, 550 wavelengths. So how can we use this in order to measure G proteins? Is that if we take, for example, G alpha, the G alpha subunit, attached to it a cyan fluorescent protein, and then the G beta gamma subunit, and attach to it a yellow fluorescent protein, then if the two are in close proximity to one another, we will have the distance will be close enough to allow this fluorescence energy transfer. And if we excite it with this laser and see a fluorescence in this wavelength, then we know that these two are in close proximity to one another. Okay, this is a bit complicated and fast, I know, but again, so under normal circumstances, let's say that these two are not connected to one another, or they're far from one another. If I excite with a light of 440 nanometer a cyan fluorescent protein, it will emit in 480 nanometers. And if I will excite, for example, with a laser of a 500 nanometer, the YFP, it will emit in 527 a fluorescent light. But if these two are in close proximity and I only excite in 440 nanometer, and I detect light in 527 nanometer, it means that these are connected or in proximity that is less than 10 nanometers from one another. Okay, so in this experiment, for example, what they did is that they measured what is the amount of yellow light or yellow fluorescence that they get when they excite with a blue light or with a 440 nanometer as a function of adding as a function of time, and this is done when adding a ligand. Okay, so you see that what happens is in the beginning there's again close proximity between these two, so you get a high signal, and when they added a ligand, they saw a drop in the level of the emitted YFP, which indicates a dissociation of these two molecular components of these two proteins. So the speed that this happens can actually help researchers and help people that study G proteins to quantify how fast this whole reaction happens. And you can do the same thing not only for the G alpha and G beta subunit, but you can also do it for example, you can put the YFP on the active receptor and the CFP on the G alpha, and then measure how long it takes for the G alpha to associate with an active receptor, and so on and so forth. And from that, we got the kinetics of the G protein pathway. So we can say that receptor conformational change happens in about 30 to 15 milliseconds. The receptor G protein interaction happens also in 30 to 50 milliseconds. Then the G protein activation remains, like the G proteins remains active for about 300 to 500 milliseconds, and the accumulation of cyclic AMP happens over very long periods of time, which is like 20 to 50 seconds. So you can see that there is very different time constants for different steps of the G protein reaction. And for comparison, if we compare this, so this is in terms of neuroscience, when we're talking about neuroscience, this falls into the category of metabotropic receptors, for example. Metabotropic receptors recept neurotransmitters, and a lot of times cause activation of G proteins. So try to compare like the accumulation. Let's say that the accumulation of cyclic AMP is like the end point of the reaction, or the end point of the signaling cascade. So this takes about 20 to 50 seconds, and if you compare it to like an opening of a channel in an ionotropic receptor, this takes like it's in the order of nanoseconds. Okay, like the time that it takes for a sodium channel to open, or other types of receptors to open, it's in the order of magnitude like of nanoseconds to milliseconds. So this whole process is much, much, much slower than what would happen if you only had like a simple receptor that opens a channel in response to binding of a ligand or a neurotransmitter. Okay? So the take-home message from this chapter is that G protein signal transduction, first of all, is just an example of many signal transduction pathways and cascades that we have in the body. And first of all, it's important to note that normally, also in biology, we see in general that every reaction has also a counter reaction. So you saw that simultaneously to the activation of the G proteins, there's also reactions that cause like desensitization of the receptors, and there's a lot of modulation on this response. But what G protein signal transduction enables the biological system is, first of all, specificity. We said that the receptors are very specific to the type of ligands that they know how to bind. Amplification of the signal, adaptation, like we saw when we saw in the example of the light, and finally, complexity, meaning that this cascade can lead to many different types of cellular responses inside the cell in response to a very simple signal. So I'll just finish this chapter with a quote from the book, is that very soon we will know the identity of all the pieces in many signal transduction pathways, but putting the puzzle together to predict cellular responses remains elusive. For instance, we can enumerate the G proteins, kinases, phosphatases, aristines, and other proteins that participate in signals from beta-adrenergic receptor in liver cells, but we are still far from being able to predict quantitatively how liver cells react over time to a given dose of adrenaline. In part, this is because complex feedback or feed-forward loops regulate the activity of multiple enzymes and other compartments in the pathway. So I like this quotation because it shows you that even when you look in the level of a single cell, or when we look at the cellular responses in a single cell, it's very, very complex. And it really, like for us, for neuroscientists, a lot of time we like to think about the that the nervous system is so complex because it has all these feedback and feed-forward loops, and every behavior is governed from activity of multiple neurons and stuff like that. But when you look deep, even in the level of a single cell, there's so much complicated dynamics and regulation that is like kind of worrying when you think about how long it will take for us to understand the entire biological system, but also what makes it interesting. So I think we'll take a break now and come back in 5 minutes to 12 for the last chapter. כן.
כאילו שיש כל כך כל הנורא ספציפיות בכל היום, מבינים אותו סייג ב-AMP, אז כאילו...
כן, זה נראה ככה, אבל זה באמת קומבינציה של הרבה מאוד פקטורים כנראה אחרים, שכאילו, שתורמים לאיך אתה באמת, כאילו זה גם תלוי באיזה סוג תא זה נקלט, ומתי זה קורה, וכנראה שהריאקציה לא קורית לבד, אלא נגיד אותו הורמון יכול להפעיל כמה סוגים של רצפטורים, ואז כאילו הקומבינציה של התגובה היא זאת שתיתן בעצם את הטון המולקולרי, כאילו. זה מאוד מורכב, כאילו, מהבחינה הזאת, אבל כן. איך חלבון פולט אוכל גל?
כן.
אז בגדול זה קורה ברמת האטומים, זאת אומרת יש מולקולות מסוימות שמסוגלות, מבחינת הסידור של האטומים שלהם והסידור של העננים האלקטרונים שלהם, זה מאפשר בעצם, מה שיכול לעלות רמות אנרגיה זה אלקטרונים בתוך אורביטלות, כאילו של האטומים שנמצאים בהם. ואז, אני לא יודע בדיוק איך זה קורה פיזיקלית, כאילו מה באמת גורם למולקולה אחת להיות פלורסנטית ואחרת לא להיות פלורסנטית, אבל בגדול זה היכולת של האלקטרונים, או של אלקטרון בתוך המולקולה הזאת, לעבור מאורביטלה, כאילו, ממצב אנרגטי, כאילו, זהו, לעבור מאורביטלה אחת לאורביטלה שנייה, ואז לשהות שם במשך זמן מסוים. זאת אומרת, זה גם לא יכול לקרות מיידית, לפעמים זה קורה, ואז אז הוא שוהה שם זמן מסוים, איזה פמטו שנייה או משהו כזה, ואז שהוא קופץ חזרה, אז הוא חייב לשחרר את האנרגיה הזאת, שבעצם המצב הזה היה מצב אנרגטי גבוה יותר, וברגע שהוא קופץ למטה, אז הוא משחרר פוטון. אבל הפוטון הזה הוא כבר בעצם, בגלל שהוא, לא יודע אם ראית שם, יש את העניין הזה של הרמות אנרגיה. אז בגלל שהוא בעצם איבד, בגלל כל מיני אלמנטים רזונטיביים ששוב אין לי מושג לגביהם, זה הכל פיזיקה, הוא בעצם איבד אנרגיה בתהליך הזה, לעומת ההפרש באנרגיה שלנו קודם, והוא בעצם יורד גם לאותה שכבה, יש גם שכבות ביניים כאלה. אז האנרגיה שיש לו כשהוא קופץ, שהוא צריך לשחרר כדי לרדת מפה לפה, יותר נמוכה תמיד, מאשר האנרגיה שהוא קלט בשביל לעלות לשכבות האלה.
אוקיי, אבל הגל הזה שרואים זה הסיבוב שהוא עושה?
הגל שרואים הגל הזה?
כן.
לא, זה פשוט ייצוג של אנרגיה. כאילו, לא יודעים נראה לי... זה פשוט אנרגיה. כאילו, הסיבה שזה גל, זה כי זה גל אלקטרומגנטי. כי כאילו, אור זה משהו, זה גל אלקטרומגנטי.
אבל כשאתה אומר, כאילו, יש לו, כאילו, קצב, משהו מהירות. גל מהיר יותר ו...
אז לא, כל הגלים מנעים במהירות האור. אבל כאילו, טוב, זה פעם חצים ממש לדברים פיזיקליים, כאילו, יש פשוט רמת אנרגיה שונות של אור. יש אור שיכול להיות באנרגיה גבוהה, נגיד אור מבחינה אולטרה סגולה.
כן, אבל זה משנה את הגלים, האורך גל יכול להיות קצר, או אורך קצר, נכון? זה משנה אותה.
נכון.
אז טוב, כיתה, אני לא...
מה?
לא, אז אוקיי.
לא, פשוט האורך גל, תחשבי שזה נגיד התדירות, מה זה האורך גל?
תדירות של מה, שהוא מסתובב?
לא.
של מה?
אז זה צריך כבר, כאילו, צריך כבר באמת לדבר על פיזיקה של אור.
כן.
ובאמת שאני לא בן אדם בשביל לשאול אותו את זה. אבל רק צריך לדעת שיש כאילו, יש אנרגיות שונות לאור שונה. ומה שחשוב הוא שאורך גל באנרגיה גבוהה נקלט, ואורך גל באנרגיה נמוכה יותר נפלט. אז פה זה אורך גל באנרגיה של 440, שהוא יותר אנרגטי. כן, סגול יותר, ואז צהוב. אוקיי?
Very much. Can you see the paper myself? Did I take images? Or was it just...
Which paper? This from the textbook.
Yeah.
I don't know. I need to find out where it is. Ah,
Oh, really?
actually.
I was thinking about, if they took images, then it would probably have been green, right? Not red, not yellow.
No, actually.
It's not quantitative, this transfer.
Oh, thank you.
It's not quantitative.
CSP will still show up blue, right?
Yeah, but it's relative because like what you want to see is the drop. Because you don't want to study absolute numbers.
Yeah, of course, they looked at the yellow light.
Yeah.
I was just wondering if they
So does
took
the
actual
vaccine,
pictures.
why can't they
Yeah.
fucking do
Yeah.
it? It's
Yeah,
like
then it would probably have been green, right?
Why?
A little more, a little less green. Because when you mix those two colors, you get green.
No, but because they have filters.
Yeah, well, of course, if they use the filter with only the yellow, then
Yeah.
you get something like this.
Yeah.
But I think
they use filters.
don't you get more information if you also get the blue levels?
No, because the blue levels you can't see because this is what you excite with. Like you always need a filter for your excitation. Because
But the fluorescence is at 490.
you mean this fluorescence?
The two fluorescence.
Yeah, they got both. For sure, they got both. Because not all, it's not 100% reaction, you know. It's not 100% like. Yeah. Yeah, probably they saw both.
I'm kind of confused by the use of the term half-life.
Yeah.
Is that
It's half-life of the response, like because because it's like there's a kinetic to the response. It's not like a step. So you have to define what is the kinetics. Most things, you know, also in biology, and you can agree in pharmacology, it looks like this, right? Like a Michaelis-Menten or something. Like there's a kinetic of the response that is like decaying in a certain way. So if you look in so let's say you have like this is the.
Oh well, not if you look at complex systems with compartments.
Yeah.
You still have the input.
Yeah.
It's
But
like in biology.
But for example, here you can say that the response, like it takes it to end, like I don't know.
We've got a zero order kinetic most of the time
Where?
in pharmacology.
Okay. So in biological system, a lot of time you have like this kinetic. So you want to, what is half-life? Half-life is like the.
Yeah, it's a zero order as well.
Yeah, it's like
if
half
you
maximum.
Draw the curve like that. But you know, there's two different types of interpretation to the half-life in this thing. If you look at, for example, the interaction or the conformational change, it could be that it's 30 milliseconds to 50 milliseconds for it to affect. You know, that half of the proteins have changed conformation.
Yeah.
Or it's like the contingent of all the receptors, and you excite them, and then you look at how long it takes for half of them to, you know.
Yeah, I don't know. I don't remember exactly how they did it here. Yeah.
And the G-protein activation is the same. Is it the time that it takes for G-protein activation to have an effect, or is it the time?
Or how long? I think it's how long it stays on average. Like how long these two elements are bound together, are separate from one another.
Yeah. And if you think about the implications, for example, we think with some metabotropic receptors, then I think I would find it more interesting to know how long it takes for the first effect to arrive. Like that's probably not the 300 milliseconds, because it would be much faster.
No, it's faster. But the effect is very prolonged. Like I think that all these responses are like the initiation and the decay. Like so it's like half.
Yeah, well, but as you said, it's a complex system because you don't eliminate the hormone, for example.
Yeah.
So you have the input and the output at the same time, which is a complex compartment model. So I don't know what the half-life exactly means there.
Yeah.
We'll probably have to read the publication.
Yeah. But in this example, it's not the same from here. For example, this is the book. This example is much simpler. Like when you see here, like they just added the ligand, and then they just.
They just see how the
fast
the G-protein scatters.
Yeah.
Basically, yeah. That's cool nonetheless. Maybe Nobel Prize.
Nobel Prize? I think there is, no?
What?
Then
Did they get one?
I'm sure someone got Nobel Prize for these things, no?
It's pretty recent.
No, but these are not.
Also
This is just a paper that I.
How old is Brett? I don't really know.
At least 20 20 years probably.
Maybe someone got for it. I think you had a question.
I have a quick question.
Yeah?
First of all, did you see my email with the paper?
Yeah, I didn't get a chance to look at it yet.
And also, is there a chance you would upload, there's an exam bank for the university?
Okay.
Is there any chance you would upload, like, previous past tests?
The thing is, I can give you like previous tests. They're not very good, like, because this is the third year that I'm teaching the course, and the tests that were before weren't that similar to my test. And my test, like, so I normally give like one example exam. I can give you like from previous years, but it won't help you very much.
Okay.
The one example exam will help you more.
Okay. When will you,
I can open it right now. Don't mind.
Can I go see it then?
What?
Are you going to send it to the group or?
It's gonna be on the Moodle.
Okay.
Quite substantial amount.
Yeah.
How long is it? And does everybody have such a thing, or did you volunteer for that?
I think you volunteer, you get money for it.
So it's separate from your...
Yeah, it's separate.
Okay, because it's included in ours, but we have different organizations. So
No. Here, some people have, some people don't.
Okay, so you just...
But it's something...
kind of organization for that, and you
Yeah.
just sign up for some teaching.
You sign up and then you're selected, and it helps also that you're...
Yeah, of course. Yeah, we have that automatically, but we do not have the entire, seminar, we just do parts of it.
Also here it's pretty rare that like a PhD student will give an entire course, but it's like a more general background course. You saw we're going through things
Like
really fast.
School.
Yeah, really fast and superficially. So it's like see, last test was too fast, but sometimes I don't control my own speed. Good. Okay, so I hope you're excited because we came to the last chapter of the excited and disappointed, that we came to the last chapter of the course, which is post-transcriptional gene control, or to put it in more simple terms, what happens to an mRNA molecule or to an RNA molecule from when it was transcribed to its functional role. And this is taken from the book, so obviously the book is old, so they say amount of specific protein produced, because I told you that 15 years ago or 10 years ago everybody thought that RNA is only good for protein making, but nowadays we know that it's not true. And also a lot of these different steps that they show here, so let's just go over them quickly. So we can we'll divide this chapter for regulations that are happening inside the nucleus and regulations that are happening in the cytosol, including, I'll give one example of what happens when the RNA molecule is moving from the nucleus to the cytosol through nuclear export. And so what happens, initially what happens to the mRNA is splicing, cleavage, polymorylation, and capping. Then you have RNA editing, which here they say that's rare. Nowadays we know it's not that rare. And in the cytosol, you have cytoplasmatic localization. Again, in the past they thought it's very rare, but nowadays we know that mRNA localization, it's not rare. Actually, in neurons, it's more a rule than an exception. And most of the, like I told you, in previous classes, most of the neuronal, strictly neuronal mRNAs are localized in the cell. It's not random. They don't come out of the nucleus and just search for a close-by ribosome for them to be translated. They're actually directly localized to specific compartments of the cell. Then the translation is being regulated, and finally, the capping and decay of the mRNA also has. So all of these are levels of regulations that we see inside the cell, and we will divide this chapter again to the nucleus and to the cytoplasm. First, we'll start by very basic processes that happens to eukaryotic mRNA, which is capping, polymorylation, splicing, and alternative splicing, and then transport, and then we'll describe cytoplasmatic mechanisms of post-transcriptional control. This is more recent and new stuff, including microRNA translation, repression, siRNA regulation, and finally mRNA degradation, which all of these have levels of regulation inside the cell. So we'll start from the beginning. And the first thing that happens to an mRNA, even while it's being transcribed, so after the RNA polymerase starts transcribing the mRNA from the DNA, when the RNA reaches about 25 to 30 nucleotides in length, special 7 methyl guanosine is added to the 5' end of the molecule. So again, remember that when RNA polymerase transcribes the DNA to RNA, the first part that comes out is the 5' end. Okay, so if I have my gene, and this is the beginning of the gene, this is the end of the gene, then when RNA polymerase binds to the DNA segment, again this is double-stranded, just so you won't get confused, it starts moving in this direction, and it starts making copies of one of the strands of the DNA in form of RNA. So in the the initial segment that comes out, or the directionality of the RNA that comes out from the from the RNA polymerase is always 5 prime. So after in the initial processes or the initial steps of transcription, you have an addition of a 5 prime cap that is added to the 5 prime end of the growing mRNA molecule. And this is done in 2 steps. One, there's an enzyme. Actually, you can divide it into 3 steps. One, there's an enzyme that removes the natural phosphate from the 5 prime end of the RNA. Another one that adds a GTP to this end. So you see, this is the GTP that's being added. And lastly, a separate enzyme that attaches a methyl group. You see, this is a methyl group to the N7 position of the guanine base of the GTP. And this capping is vital for mRNA stability. For example, mRNAs that do not have capping or are not capped are rapidly degraded in the cell. The second type of processing happens in the end. So after we have our capped mRNA molecule, I told you that one of the mysteries of eukaryotic transcription in general is that we don't know, like the RNA polymerase can just keep going. We don't know where it stops. It doesn't have a clear stopping signal. For example, it can, like one time when you transcribe a gene, the molecule that you can get, the pre-mRNA can be this length, and the other time it can be like this length, and the third time it can be that length. Okay? But in the end, what we see is that we get more or less the same mRNAs. And one of the reasons for that is the addition of a poly-A tail, or polyadenylation tail, which is dependent on a polyadenylation signal, which is normally AAU, AAA, but there are other signals also. And this signal is always, like, it's in the same position. And there are specific enzymes that know how to identify this signal, bind to it, then cleave the mRNA. That doesn't actually happen in the end completely, but they know how to cleave the mRNA right after this segment, and then separate enzymes add a poly-A tail to these molecules. So even if we don't really know what is the mechanisms or what happens or why we get different lengths of mRNA, in the end what you get is the same molecule. Even if the RNA polymerase made initially a molecule that's much longer, what you will get right after the initial transcription is the mRNA that will always have more or less the same length and will have a long polyadenylation tail. So beside the fact that it cleaves the mRNA molecule to the same length each time, it also provides very important, again, stability. So this poly-A tail also protects the molecules from degradation, and it also has a very important role in promoting of the translation of the molecule. Okay, so both the 5 prime cap and the 3 prime poly-A tail have roles in the regulation, first of all, in prevention of degradation by what we call exonucleases. So exonucleases are special enzymes that, you can say, eat the mRNA from either side. So they protect from degradation from exonucleases that degrade RNA from either the 3 prime end or the 5 prime end. They promote, like these 2 elements are very important for translation, because we didn't really get into the details of translation, but you can imagine that it's more complicated than what we learned. But one of the things that's important for translation is that when an mRNA is being transcribed, when it's being translated, it has to form this loop, and this loop is mediated by enzymes that know how to bind the 5 prime end, and in the end they also bind to the poly-A tail. So if a molecule doesn't have a poly-A tail, doesn't have a 5 prime cap, it will not be translated in general. Okay, everything that I'm saying has an exception, obviously, but if we're looking at classical molecular biology, then they have to have association of both of these to recruit the center protein that will then recruit the small subunit of the ribosome that will recruit the large subunit and start the translation of the mRNA. Okay, if one of these elements is missing, it won't happen. And lastly, these are also important for regulation of nuclear export. So again, mRNA molecules that don't have a poly A tail or don't have a 5' cap will often not get exported from the nucleus. Okay, so the next thing that happened after the adding of the, actually it's not completely correct what I'm telling you. It's not like, sometimes this process that is called splicing happens simultaneously to the, like even before the mRNA is completely transcribed, splicing can occur. But in general, after the 5' cap is added and the poly A tail is added, then you have splicing. And splicing is the editing of the mRNA. And like I didn't put here a picture of a gene, but I hope that you remember that we said that the gene is divided into segments that we call exons. And actually, it's not a good color. I don't want to confuse you. What? So we can divide each gene into segments. What do you prefer, green or blue? Blue? Okay, so blue will be the exons. Okay? So we can divide each gene into elements that we say that are exons and elements that we say that are introns. Introns and exons, like their name. Exons are elements that from the original DNA that will make it to the nuclear export, that will remain in the mature mRNA molecule when it's exported from the nucleus. And introns are segments that will remain inside the nucleus or get degraded or get removed from the molecule in the end. So the process of removing introns is called splicing. And splicing is basically attachment of segments of exons together to form the spliced or the spliced mature mRNA that will later be exported from the nucleus. Okay, and this happens through, this is a massive oversimplification, but still it happens through two steps. First, somewhere along the intron you have this A base, and this A base attaches and forms like a lariat or this loop with the GU here, and then lastly these two segments are attached, and this lariat is removed. And the consensus sequence for that is surprisingly very permissive. Okay, so you don't need like a very strict consensus sequence to determine what is an intron and what is an exon. And again, I remind you, there is no difference between an exon and an intron in terms like of the base pair or the bases that it contains, stuff like that. It's exactly the same. It's like a piece of RNA. But still, there are what we call a consensus sequence that mark the locations of these junctions between the exons and the introns. Okay, and this is, if we look at a typical pre-mRNA, this is the frequency of occurrence, meaning that what is the frequency that we see in a splice site or in this junction between an intron and an exon, like the identity of the bases. So in 100% of the cases, you will always have a GU in the beginning of an intron, and you will always have an AG at the end of an intron. And normally, you will have also this A that will be located somewhere inside the intron itself, and also other types of elements, but they're less important. And but there's much of the center position of the introns that don't really matter what you have inside the intron. These two brackets can signify like thousands of base pairs. Like introns are very, very long. In eukaryotic genes, it's very typical for 90% of the genetic material to be removed in this process of splicing. So, and it's kind of surprising that these very simple consensus sequence provide the information for the cell to know where to splice. Okay, because it's just like GU, AG, and an A somewhere along the sequence. So the proteins or the RNA molecules that do the work of the splicing are in general termed nascent RNA, or the the nascent RNA, or the RNA in the cell that is being that will be processed is done. The processing is done by molecules that we call them H and R and P. And this is, for example, an antibody, like a staining for these H and R and P. So you can see this is an mRNA molecule. Oh, sorry, this is a DNA, and there are mRNAs that are springing from this DNA molecule. But this is staining for the H and R and P. So you can imagine that in a given cell, the RNA is not naked. Okay, it's covered with these H and R and Ps, which are proteins, and they're proteins that are called heterogeneous ribonuclear protein particles. So these nuclear particles contain proteins, and they also contain, and they associate to small ribonuclear RNA. Okay, so they're composed both from protein and from RNA. One of the examples of this short RNA or small nuclear RNA molecules that are in charge of the splicing, that inside the splicing complex they're called uRNA, and they're short RNA molecules at about 150 nucleotides, and they facilitate the processing or the cleavage of mRNA, including splicing, and they do it by mediating between the RNA and the proteins that have to cleave, for example, the RNA. And this is like an example of U1 siRNA that is responsible for identifying the junction between the exon and the intron in the 5' end, and U2 siRNA that is responsible for identifying the branch point. And there are other uRNAs or small RNAs that are responsible for the rest of the function of splicing. So and here we look like specifically what happens in the process of splicing, and one of the most I can say like amazing phenomena with splicing, it is that interestingly it doesn't require energy. Okay, so we said that in almost all the reactions that we learned about in class, it always has like energy that's coming from either GTP or ATP or phosphorylation or something that like provides like molecular energy. But in splicing, this entire process does not require energy, which is a little bit strange and also interesting. And this is one of the reasons, for example, that people think that RNA. I told you that that RNA has the ability for the enzymatic activity, and has the ability to create structures and to create secondary structures, and to the most important enzyme in our body, which is the ribosome, is composed of many RNA molecules. And one of the reasons that researchers think that RNA preceded DNA is that it has this amazing capability to spontaneously create structures. So it has the ability, this RNA molecule, has the ability to like cut and paste itself, and sometimes it happens in some introns. In some cases, it can happen by itself without the mediation of any proteins or external RNA molecules. So it has the ability to like cleave itself and remove elements from itself without the requirement of external energy, which is for scientists, this is one of the things that gives this intuition that this is something very ancient in terms of evolution. Like these processes are very ancient.
So what's the trigger?
What?
What is the trigger?
What's the trigger for splicing like that? the trigger for splicing here is just the cascade of that the RNA is made. So the RNA is made, and then first of all, in eukaryotic cells, this is highly regulated, this process, with H and RNPs and the uRNAs, like you're going to see in a movie in a second. But but we can imagine that, like in their early life or the early molecular life, this could happen potentially without the need of an organism at all. Okay, this, like if the RNA molecule is just floating in the ocean, it can potentially like cleave itself and make new structures spontaneously without any energy. Okay, so and this reaction, or the first, the breaking of these of this bond in the creation of new phosphodiester bond is called transesterification. And for sure, by now you remember that the bond that holds nucleotides or DNA or RNA molecules, it's called phosphodiester bond. So the reaction in which one phosphodiester bond is switched by another is called transesterification. Okay, and it happens again. You can see here molecularly how this oxygen, like first, the initial step is the attack of this OH of this phosphate, and then this oxygen attacks this phosphate, and what leads to the cleavage of this lariate out and the connection of these two segments together. So a spliceosome is a general word for a complex that in eukaryotes, again, like in complex life forms like us, is a very, very intricate and complex machinery. It involves about 70 proteins and RNA molecules that are involved in this machinery, and the goal of this machinery is to mediate proper splicing. So, although I told you that like the mRNA has the capability to splice itself, effectively in eukaryotic cells it doesn't happen by itself, and it's mediated by a large amount of proteins and a lot of like regulation steps that mediate this spliceosome, or the splicing of mRNA. And again, so you won't have to imagine too much. You have a movie.
Imagine you are hiking through the woods on a late summer's eve when you have the misfortune of coming across a rather disgruntled grizzly bear.
You can test your attention. Okay.
The mature transcript for many genes is encoded in a discontinuous manner as a series of discrete exons. Exons are separated from each other along the DNA strand by non-coding introns. mRNAs, RNAs, and tRNAs can all contain these encoded introns, which must be removed from precursor RNAs to produce functional RNAs. Most introns do not themselves contain genes and are simply removed from the precursor RNAs and degraded. There are a number of different classes of intron. Some are excised from the primary transcript by proteins, some by ribonucleoproteins, and some excised themselves. Yet all use a series of transesterification reactions to accomplish the task. In all splicing reactions, the two exons that surround an intron are not directly joined to one another in a single step. Instead, a two-step reaction occurs in which the intron is first detached from exon 1, freeing this exon to react with exon 2. Both of these reaction steps are transesterifications in which a single phosphodiester bond between two nucleotides is broken and replaced with an energetically equivalent phosphodiester bond without net input of energy. We will now focus on the splicing of eukaryotic mRNAs. Introns can account for as much as 90% of the length of a typical precursor mRNA. Splicing of eukaryotic mRNA is catalyzed by the spliceosome, a large ribonucleoprotein machine. The spliceosome is composed of 5 individual small nuclear ribonucleoproteins, pronounced SNRPs, which comprise both RNA and protein components. These SNRPs are U1, U2, U4, U5, and U6. Each one contains an RNA molecule called an snRNA that is usually 100 to 300 nucleotides long, together with several protein factors. The snRNAs perform many of the spliceosome's mRNA recognition events by forming Watson-Crick base pairs with the precursor mRNA and with one another. The protein factors recognize specific sequences in the mRNA or help to promote conformational rearrangements in the spliceosome that are required for the splicing reaction to progress. Before splicing can occur, the spliceosome must identify the splice sites, the sites at which exons are separated from the neighbouring introns and at which two exons will be subsequently attached. The spliceosome identifies splice sites in part by recognizing extremely short sequence motifs found in each pre-mRNA. These motifs are a GU sequence at the 5' end of the intron and an AG sequence at the 3' end of the intron. In addition to these sequences, a branch point nucleotide within the intron and a polypyrimidine tract upstream of the 3' splice site are key elements that are recognized by the splicing machinery. We also understand that there are general features of exonic and intronic sequences which result in the tendency of certain proteins, like the SR proteins, to bind to exons and other proteins, like the HNRNP proteins, to bind introns. These differences also help to define exons and introns. The first step in the splicing reaction is the recognition of the 5' splice site by the U1 SNRP. Other splice site consensus sequences are recognized by non-SNRP factors. The branch point sequence is recognized by the branch point binding protein, BBP, and the polypyrimidine tract and 3' splice site are bound by two specific protein components of a splicing complex referred to as U2 auxiliary factors, or U2AFs. U2AF65 binds to the polypyrimidine tract, and U2AF35 binds to the 3' splice site. Following the binding of these initial components, the remainder of the splicing apparatus assembles around them, in some cases displacing some of the previously bound components. Next, the precursor mRNA undergoes a structural rearrangement and the first transesterification step takes place. During this step, the 2' OH of an adenosine residue located within the intron attacks the exon 1 intron boundary, detaching the intron from the exon and producing a branched intron structure called a lariat. Other rearrangements in the spliceosome then bring together the newly freed exon 1 and the intron exon 2 junction. The terminal 3' OH of the newly released exon attacks the intron exon 2 junction. This second transesterification reaction completes exon ligation or splicing, releasing the lariat intron. While the molecular details of the individual steps are not fully understood, it's clear that the spliceosome is a dynamic ribonucleoprotein machine, and that conformational changes in the RNA components themselves are central to the spliceosome's function.
Okay. Okay, so for example, one of the things that I told you is that because this consensus sequence is not very like a little redundant, and when we think about it in introns that are very large in size, you can have these sequences occurring randomly along the segment of the intron. There's a second type of proteins which are called SR proteins, which are probably important for these proteins specifically bind to the exons, and also many researchers think that these proteins, because like I told you, there are proteins that are covering proteins in small RNA molecules that are covering the entire mRNA. So these SR proteins are like marking the area of the exon. So instead of like scanning where along the intron is these like splice site or these junctions that need to be spliced, the cell probably tags the exons, which are much shorter normally than the introns, with special type of proteins that protect it from the identification as an intron. And these SR proteins also interact with the U elements of the spliceosome. So in the end of the SR proteins, when there are no SR proteins, the U1 or the other U proteins can bind and initiate the splicing machinery.
So if you have a sequence of a gene, then you can't know what the other exons of the other exons because you don't know what the potential binding.
If you have a sequence of a gene,
It's just a sequence.
yeah,
You can't know where is the exons of the other exons.
it's often very hard to predict, like bioinformatically, from the sequence. Yes, you're right that bioinformatically it's very hard to predict what is the sequence of the exons and the introns. Often this is determined experimentally, and not only that, is that your question beautifully leads me to the concept of alternative splicing, in that in different types of cells, or also under different conditions, we see that sometimes an exon is an intron, and sometimes an intron is an exon. Okay? So the fact that under certain conditions, for example, if we take the same cell and put it, for example, under stressful conditions, this can trigger a change in the way that the cell will interpret which one of the sequences of the gene will be considered an exon and which one of them will be considered an intron. And this whole concept is called alternative splicing. And in general, in humans specifically, 75%, and probably this is more by now, of all the genes in our genome are what we call alternatively spliced, meaning that one gene can potentially create multiple variants of a protein. Okay, and certain elements can be present or deleted under different conditions. And the most extreme example of that is the DSCAM protein, which is short for Down syndrome cell adhesion molecule, that is highly expressed in the development of the brain. And again, analysis of this gene, of the expression of this gene, found out that there are 95 alternatively spliced exons, meaning that either one of them can be inserted, excluded, or included in the mRNA, in the mature mRNA molecules, like in different conditions or in different cells, which gives you 38,000 possible isoforms of a single gene. So when you think about it, our human genome, in the human genome, you have 20 to 25,000 genes, but in a single gene, you have the possibility to create more isoforms than the genes that you have. And in Down syndrome, where this gene was discovered, it's overexpressed because it's included in the chromosome 21, which is the trisomy in Down syndrome. And it's believed that this cell might be this protein might be very important for the connection, or the fact that it's called adhesion or cell adhesion molecule, is that researchers have seen that this protein is expressed on the surface of the cells, and it might play a very crucial role in the recognition, for example, of two neurons. Because one of the things that we don't know is why two neurons connect to one another and not other ones. And this might be one of the answers to this question, or at least part of the recognition sequence that is important for two neurons to recognize each other and form the right connections. And also, and because of that, it's also easy to understand why, if you have a overexpression or an abnormal expression of this gene, it can lead to wrong connectivity or impaired connectivity between different neurons in the brain, and cause all sorts of syndromes, including Down syndrome. A second example of alternative splicing, which you would never think about before, is how alternative splicing changes the activity of hair cells in your cochlea. So the cochlea is the organ in the ear that is responsible of hearing, and this is like the cochlea that is spread out. Normally it's like in this looped structure, like a snail. But this is a special type of membrane in which in one type of the membrane it's thicker, and one side of the membrane is thinner. And the physical properties of the membrane determine are encoding frequencies of sound from the different spectrums. For example, in the thick portions, these hair cells will vibrate at 50 hertz, and in the thin portions, the hair cells that are responsible for detecting this vibration will vibrate at 5,000 hertz. Okay, but beside this specificity, beside this tonotopic organization of the organ, there is also a level of fine-tuning of the hair cells, or the sensitivity of the hair cells is determined by their sensitivity of calcium. Okay, and for example, this so the calcium concentration the calcium concentrations that open a specific type of potassium channel is determined, or the sensitivity for the calcium concentration that will determine a potassium channel is much determined by the alternative splicing variants of that channel. So, for example, if you have a channel that includes, so this is like this shows you like illustration of this potassium channel, and this potassium channel has certain regions in the protein that can be modulated by alternative splicing. So, in different types of hair cells, you can have different variants of this channel that will be expressed, and hence you will have different sensitivities of this potassium channels to different levels of calcium concentration. And people think that this is like one of the ways, for example, that besides this rough tuning that is determined by the thickness of this membrane that is felt by these hair cells, the activity of the hair cells themselves, or the sensitivity of the hair cells themselves, is determined by the combinations of different splice forms or different splice variants of this calcium-dependent potassium channel. Okay, so in general, in these 8 splice sites, you can get a total of 576 different combinations, and each one of these combinations has different sensitivity to calcium, and this can also change during the life of this hair cell. Yeah.
576 is a statistical calculation, or they were found in?
I think it's just a statistical calculation of like the different combinations, because not all combinations can exist together. So it's like theoretical. I don't believe that they really looked and found 576. Well, it's possible, but not likely. Probably it's just like a theoretical bound. Questions? So splicing, or one of the splice sites, is sometimes suppressed. What people have seen in research is that the thing that determines whether a specific splice variant will be formed is often due to the activation of a specific protein kinase that is activated by depolarization. Okay, so if we can turn back to the chapter about G-proteins, you can imagine that if the cell receives, for example, a significant amount of depolarization, it can, for example, activate pKa in a high amount, and this activation of pKa will cause not like activation of a transcription factor and generation of a mRNA, but it can cause also alternative splicing. Okay, so and this alternative splicing will cause the entire cell, for example, to be less sensitive or more sensitive to the concentration of calcium, thus fine-tuning the cell to a specific frequency according to the activity of the cell. So it's like a very complex molecular feedback loop that goes, that sits on the regulation on the level of splicing. Questions about that? Because it's pretty complex. I know. So this is the estimated, like a piece of history. This is an interesting piece of history in terms of how many genes once were thought that the human genome has. Okay, so like in the past, not so long ago, like in the 90s, it was estimated that the human, like in 1964, it was estimated that human gene numbers were like 6 million genes. Then it started decaying, and now it's pretty much agreed that we have about 20,000 protein coding genes. Let's say that, because people were trying to find something that is special about humans, like what is like unique about humans that's different between us and other animals. And actually, when you look at alternative splicing, so before that, when we look at the number of genes, you find out that a grape has much more genes or protein coding genes than humans, and like chicken has a little less protein coding genes than humans. So this is probably not, like the number of genes is probably not the right way or the right place to look for any differences. And again, everything that I'm telling you is very speculative, so don't take it literally. But if we try to estimate the amount or the percent of alternatively spliced exons, we found that the humans have the most or the highest percent of alternatively spliced exons, so meaning that genes that can undergo alternative forms. And specifically, in the brain, out of all the other tissues, you have the most, the highest proportion of alternatively spliced genes. And in general, I think nowadays it's pretty much agreed that probably the differences between us and other organisms is in the level of regulation. Okay, so I'm not saying alternative splicing by itself, but alternative splicing is like an indication that shows you the complexity of regulation that, for example, a gene can have. And when you look at these levels of regulation, you find out that humans have the highest levels of regulation, and specifically the brain. But again, it doesn't mean that much, because you see the liver is not that different. So I don't know if it's very convincing. A specific example that was found in our lab, 16 years ago, is that acetylcholinesterase, which is the enzyme that is responsible for breaking down of acetylcholine in the synaptic cleft, under stressful conditions, has a tendency to change the variant, or has alternative splicing that changes from a normal variant to an alternative variant. And this alternative variant is actually in contrast to the normal synaptic ACHE that is bound to the synaptic cleft. This alternative form of ACHE tends to not be, is not bound to the synaptic cleft, and is more diffused in nature, and we believe that it's much less efficient in breaking down of the acetylcholinesterase. And this happens, for example, this is a staining of the RNA. In this case, it's not staining of protein, but staining of RNA. That specifically, this alternative form of the RNA, that in mice, in the hippocampus of mice, under stressful condition, after stress, you see a high expression of this form relative to baseline. So this is one of the interesting examples in which this read-through, or this special variant of this acetylcholinesterase, was not seen before in any case. Okay, it was not seen in different cell types and other organisms. It was only found under stressful conditions. So not only so the alternative splicing is not only restricted to different cell types, but it's also under different cellular conditions. So I'm thinking about, let's just, because it's very short, we'll just go over the macromolecular transport across the nuclear envelope, and then we'll finish. And next class we'll finish the cytoplasmatic part. So this is the nuclear pore, and this is like a electron microscope image of the. This is like the external the external part of the nuclear membrane. You remember that the nucleus of the cell has this double membrane that's surrounding it, and to cross this double membrane, you have these special core complexes. That on one side they have this cytoplasmatic filament, like you can see here, and the other side they have this nuclear basket. But basically, it's a huge complex that's made of many types of proteins, and most of them are in charge of identifying or acting like as a gateway or bouncers, or like security agents to make sure what is entering the nucleus and what is exiting the nucleus. And in terms of mRNA processing, if you remember the article that we talked about by Kandel, one of the things that they did is that they added an artificial intron in their construct, and we said that it looks a little bit strange because why just add an element that has no function? And I told you that this is kind of a way to show the cell, or in the logic of the researchers and also today, but today we have better ways to make sure that the RNA will be stable. But in the past it was thought, and it was probably true, that the cell the cell knows that if some mRNA molecules undergo processing, then it's probably okay. Okay, and if it finds, for example, an mRNA molecule that didn't go through splicing, it will probably not let it exit the nucleus. And one of the reasons to do that is that the place where the junctions were, or the place where the junctions between the exons and the introns were, special types of RNP proteins remain bound to the mRNA and signal for this complex that it's okay for the mRNA to pass or bind to this complex and mediate the transition of the mRNA from the nucleoplasm to the cytoplasm of the cell. Okay, so next class we're going to finish the last part of the chapter, which is the cytoplasmic mechanism of post-transcriptional control, and then we'll go over the things for the test and things for the assignment and any question that you might have. I told you that normally this year, because of Christmas, normally I would finish the material in this class, and then you would have 2 weeks to prepare for the exam. But still, you can start preparing from now to the exam. I will put also an example test online in the Moodle, and any questions that you might have, start collecting them from now, and we'll have time to go over them next class, because we need half an hour to finish the material, or 40 minutes to finish the material, and then we can take the rest of the time to answer questions. Okay?
Makeup of it. If we don't have a background in chemistry.
What?
If we don't have a background in chemistry, how much should we use to review the chemical makeup of different, or should we just know what they do?
You don't need to know.
I don't need to memorize what the.
No, you need to know, for example, that in the DNA you have a bond that's called phosphodiester bond.
Right.
But you don't have to know
exactly
the phosphate, oxygen, and stuff like that.
Okay, so I don't have to worry about that. Okay. Thank you so much. Do we have to know all the numbers of the different ones?
All the numbers,
just
yeah.
the letters?
I found out because I'm not sleeping well because of the baby.
Yeah.
I found out that the thing that gets affected most is my speaking.
Yeah.
That I like.
Non-mother tongue speaking is very.
It's very. Yeah.
Attentional control.
No, it's really when I try to think about something and talk in the same time.
Yeah, it's basically the same thing that I experienced short term because of the hospital, because it's so noisy there.